Good afternoon, everyone. I hope that you guys are having a great start to your Thursday. Today, we are working on and going to be talking about catalytic mechanisms. So, we've been talking about enzymes. Most recently, we talked about glycolysis. And now, what we're going to do is we're going to be looking at how do individual enzymes make and break bonds. We showed one of the enzymes that we looked at last time was phosphofructokinase. Could anyone add to the chat? Maybe, let's see, phosphofructokinase 1. How about tell me the product? And you can just go with the letters, the acronym, the product of phosphofructokinase 1. So phosphofructokinase 1, yes, fructose 1,6 bisphosphate. Phosphofructokinase 1 phosphorylates fructose 6 phosphate. So it's a transferase. It adds a phosphate from ATP. Fantastic. Now, what we're going to be doing today is we're going to be taking the class or the general idea of an enzyme, and we're going to be looking at, well, how do the different amino acids that make up an enzyme actually push electrons around? So that's our objective for today. And we're going to get into that. We're going to use a couple of examples. I'm going to introduce you to a couple of different enzymes. And we're also going to wrap up with talking about how enzymes can be regulated. So, catalytic mechanisms. Enzyme reactions are described in the same way as non-catalyzed reactions. And so, if we were to look at something like this right here, where we have some electrons. Oops, sorry, I rearranged my computer desktop. If we look at the electrons on an amine, oh, sorry, the slide deck that I'm using is titled Week 8, Day 2, Enzyme Mechanisms. Week 8, Day 2. And actually, let me go ahead and I've got our Canvas page open. I will grab that link and share it with you guys. Copy link address. Done. That should take you directly to it. Thank you for asking. Okay, so catalytic mechanisms. We've got some electrons on this amine. I always like to think about partial charges. I always like to think about why is one group attracted to another group. We've got a partial negative charge on this amine. We've got a partial positive charge on this aldehyde or this ketone. Either way, this carbonyl carbon. And what we're able to do is share those electrons, making a bond between them. And we have this sort of imine or this shift base in the process. And then ultimately we have some electrons push off so that we can produce or release a group with the help of a proton. We make some water out of it. Okay, great. Now, the pushing of electrons, probably isn't too radical of an idea. I remember doing that in Organic 1. And then I know that this class has a co-requisite of Organic 2. So, or if you've taken Organic 2, moving electrons around, not a wild idea. Now, the difference between this and, say, your organic class and ways that you've looked at mechanisms historically is here what we're doing is we're using different amino acid R groups as like the sources of some of those electrons. So our three basic types of enzyme mechanisms are acid-base catalysis, covalent catalysis, and metal ion catalysis. In acid-base catalysis, it's pretty simple. We're dealing with an acid and a base. We're looking at protons being added to a group or being released from a group. Covalent catalysis, we're focused on making a covalent bond. And last but not least is metal ion catalysis. We are using a metal ion in order to move electrons around or to stabilize a group. The first one that we're going to get after is acid-base catalysis. And what we're going to do, just so you guys can kind of follow the general structure, is we're going to look at each one of these different things, acid-base catalysis, covalent, and metal ion catalysis, in a general sense. I'll give you the rundown of what's taking place, and then we're going to look at some specific mechanisms. So we'll we'll do it in that order. Um, Acid-base catalysis is involved in all enzyme reactions involving proton transfer. So if you see only a proton being moved, think about it as, okay, acid-base catalysis is involved there. Now, with that in mind, there are enzyme mechanisms that use multiple different types of catalysis. So it might be classified as covalent catalysis, but there is an element or an aspect of it that is acid-base catalysis. So what we are looking for is a proton being transferred, yes or no? As simple as that. If the answer is yes, well then acid-base catalysis is involved there. Now, general acid catalysis. In general acid catalysis, a general acid donates a proton to a substrate. So think about that as we know about the enzyme active site. So those are the amino acid residues that are present and that are involved in actually catalyzing the reaction. If a mechanism follows general acid catalysis, one of those residues must be protonated. It absolutely must be protonated. Now, the opposite of that is general base catalysis. Well, a general base accepts a proton. Um, Before I fill in this next spot, and hopefully, and if you're not looking at this slide deck at the moment, what do you think the state of an active site residue is in general base catalysis? Put a P for protonated or a D for deprotonated. What do you think? Yeah, it's going to be deprotonated. It's the exact opposite. When it's deprotonated or when something is deprotonated, it can act as a base. So I want you to think about that and start using that terminology if you're not yet doing so. Think about something acting as an acid, something acting as a base. If something is acting as an acid, that means it has a proton that it can donate. If something is acting as a base, that means it does not have a proton but will gladly accept it. So, when you think about amino acids, are there any particular amino acids that stand out to you as, oh, that could probably act as an acid or a base? Put the single letter code for an amino acid that could probably act as an acid or a base. Put that single letter code in the chat. So, it is either going to be potentially protonated or deprotonated. I'm seeing the two most recent letters that I put the two most recent single letters that I see in the chat are D, which is either a response to my previous question about deprotonated, or D for aspartic acid. Aspartic acid is an amino acid that could act as a base because typically at a neutral pH, it's going to be deprotonated. What about an amino acid like leucine? Do you think that leucine could act as an acid or a base? Add a Y or an N to the chat. If it can act as an acid, put a Y, N for no. You're absolutely right. Leucine cannot act as an acid or a base. What about lysine with the single letter code of K? Put a K in the chat if lysine could act as an acid, potentially, or, under the right circumstances, maybe a base. Yeah, because lysine is an amino acid that at a neutral or a physiological pH, it's going to be protonated. So if something's protonated, that means it can act as an acid. If something, yeah. Okay. So in general base catalysis, an active site residue is going to be deprotonated. Now, concerted acid-base catalysis, this is the type that I want you guys to remember, because this is the type that we're going to explore specifically. A general acid and a general base both participate in the reaction. So that means you've got one amino acid that is protonated and then one amino acid that is deprotonated. I'm putting a capital letter P for protonated and a D for deprotonated. All polar positive residues? Yes, Melanie, that's a great question, great observation. Your polar positive residues, chances are they're going to act very well as acids. And then your polar negatives are going to act very well as bases. Now, specific or to take a more comprehensive look, the amino acids that are most important and therefore kind of your most commonly observed in acid-base catalysis are, well, here's our polar negative, P negative. Here's two of our polar positives, lysine and arginine. Cysteine, well, that with that thiol, that can act as a proton donor. It can act as an acid, and sometimes it can act as a base. Histidine, oh man, histidine we classify as a polar positive, but one of the things that you see on your test, but also I mentioned this one before, and I think that it's good biochemical trivial knowledge. It has a pKa of about 6.1. So that means if you're at a neutral pH, more than likely it's going to be deprotonated, but you might have some that's protonated. So what that means is that histidine at a pH of about 7, you could have two histidines acting in different ways. One acting as an acid, one acting as a base because you're so close to that pKa. Could everyone give me a 7 in the chat if you follow that idea about histidine? And if that doesn't make sense to you, don't hesitate to mention it. We're going to look at that in just a little while. Two other residues that can act as acids or bases are serine and tyrosine. They both have these OH groups, and they can act as acids or bases, respectively. And so I'll just go from there. Basically, I'm not really going to ask the sorts of questions that I ask about this really are more like, hey, here's the residue. This is what it looks like whenever the reaction starts. Is that acting as an acid or a base? I don't want you to be able to identify that. The more that you remember about your amino acid structures, the better off. But you can also kind of use context clues. Context clues. Sure. So histidine has this R group right here. This imidazole ring has a pKa of about 6.1. Now, most living systems, like most of your cells, most living organisms have a pH in their cells of roughly 7. And if you remember, the buffering range of a molecule is plus or minus 1 unit from the pKa. So what that means is that at a pH of 7, you've got some of the molecule that looks like this. The majority of it is going to look like this, but you've got a mixture. You kind of got like 95, maybe as much as 99% of the molecule looks like this and 1% looks like this. But what that means is that you got 1% that looks like an acid, or that could act as an acid, and then about 99% that could act as a base. So you've got a mixture of those two forms. So in any given cell, histidine within an enzyme can act as an acid or a base. Does that help the explanation? Or sorry, was that and okay, awesome. Thank you very much. So one of the things that I want you to take away from this, and this isn't so much like, oh, I need to memorize this graph, but think about this graph as your y-axis being 0 and then 0 to 100. Your x-axis being a pH range from a low pH all the way to a high pH of 14. On the same thing for this graph, y-axis is 0 to 100, and then the x-axis ranges from 0 to 14. What this is showing is that if you have an enzyme where an active site residue is acting at a base, chances are that enzyme is going to have a higher activity. We're looking at the y-axis, a higher activity. Notice how the activity increases. A higher activity, higher ends activity at higher pH. First of all, could you give me a 6 if that makes sense? Okay. Now, the exact opposite is going to be true for a general acid. So if you have an enzyme that is dependent on an amino acid being protonated, well, you're going to have a higher activity, higher enzyme activity at a lower pH. Or another way of putting that is a more acidic pH. Now, truth be told, more times than not, you might have an enzyme that has elements of an acid and elements of a base, or residues that need to be protonated and need to be deprotonated, in which case, at a low pH, you're not going to have a whole lot of activity. And then at the kind of opposite end of that, at that equally high pH, you're also going to have a low activity. So every enzyme is going to have this sweet spot pH range where it has the most activity. And it all depends on, well, what is that amino acid, or what do the different amino acids in the active site have to look like? Could everyone give me a 1 to 3? A 1, I feel good about this. A 3 is, I'm looking forward to seeing the example. I think that will help. And then a 5 as who? 1 is I feel good. A 3 is I'm looking forward to seeing the example. 5 is who is enzyme? Nice. Okay. Okay. I think once we look at the enzyme, that's going to help out a whole lot. And actually, what I'm going to do, I know I lied a moment ago, and I apologize for that. I'm going to go ahead straight to the example, so then we'll circle back and talk about the others. Just, I think that will be helpful. Okay, so here is the ribonuclease A enzyme mechanism. Now, if you look at this, you see 3 snapshots. Upper left, right here, then right here, and then right here. Now, one thing that I want you to know, whenever you're looking at an enzyme mechanism, I want you to think about it as it's circular, in the sense that whatever your starting point is, that's also going to effectively be your ending point. Because what is one of the benefits of a biological catalyst? Well, a biological catalyst can catalyze a reaction and then kind of regenerate or recharge itself. Now, that's the first thing that I want you to think about anytime you look at an enzyme mechanism. The next thing is, I want you to think about, okay, when we're dealing with an enzyme catalyzed reaction, we have an enzyme, which our enzyme is made up of a bunch of amino acids as a protein, and then we have a substrate. Based on the name ribonuclease, could anyone guess what is the substrate? What is the thing that we act on, that our enzyme acts on? RNA. I love it. Perfect. So we're going to look at our first model. And whenever you look at your first model and you're looking at, okay, well, you need to be able to distinguish what is my enzyme? What are the amino acids that make up my enzyme? The active site residues that are important, and then what's my substrate? Now, it's a little bit tricky to see with this one because there's so much activity going on. But in the middle of this cell, or this, three-quarter circle, we've got our RNA molecule. Now, I've drawn over some arrows there, but that's all right. I'm going to erase the big circle that I just put down. That is our substrate right there. So that is the molecule that is being acted on. That is the molecule where we're going to have some bonds break. Now, did you have to know what an RNA molecule looked like in order to answer this? It helped, but we haven't gotten there at this point in this class. In order to distinguish your enzyme from your substrate, what you really need to do, and maybe what would be most beneficial if you don't know what RNA looks like, is to to look for something that identifies your amino acids? Well, his. Ooh, that's the three-letter code for histidine. Now, looking at that, I want you to notice that we've got two amino acid residues, his 12 and his 119. What I'm going to do for just a moment is I am going to use my red pen to scribble out some of the stuff. And then I want you to tell me his number 12, this one right here. Is it P protonated or D deprotonated in this first shot? Perfect. It's deprotonated. Now, his 119. Let's ask the same question. Is that P protonated or D deprotonated? Perfect. On a scale of 1 to 10, 1 being, yeah, I could identify that pretty darn easily, and 10 being, who's histidine? Where would you rank that? Okay, so this is one of the points where it's like good to know your amino acid structures, good to know those R groups. I saw lots of ones in there and I saw some other higher numbers. So that's a place that you can think about, okay, I need to be able to do this. This is the application of that skill. Now, with that in mind, our histidine 12, most people said that that was deprotonated. You're absolutely right. This is a deprotonated amino acid. Based on what we looked at previously, well, a deprotonated active site residue is going to be acting as a base. Now, the other residue in question, histidine 119. Histidine 119, if you look closely, that's protonated, and lots of people said that that was a protonated residue. A protonated active site residue is going to act as an acid. So when we go back and we think about that definition of general acid catalysis, general base catalysis, well, do we have general acid catalysis? Yes, we do. Do we have general base catalysis? Yes, we do. So what that means is that we are looking at is, oh, wrong way, wrong thing that I wanted to do. We are looking at concerted acid-base catalysis, a concert of both acid catalysis and base catalysis. We've got one residue that's going to act as an acid, one residue that acts as a base. Okay, so now back to where we were. There we go. Okay, so we've got one residue that's acting as a base, and that residue that's acting as a base, that histidine number 12, what it is going to do, it's going to pick up a proton. And it is going to look to the substrate and say, all right, substrate, do you have a proton that I could swipe? And the substrate's going to say, well, I do. And it's right here at this position. And histidine 12 says, that's great. I'm going to take that. I'm going to use this lone pair of electrons to pick up that proton. Then the electrons that were making the bond between that hydrogen and this oxygen, they're going to swing down here to another part of our substrate. And that's going to open up the opportunity for another pair of electrons to swing down and pick up this proton on our protonated residue. So we have kind of a domino type of effect. We've got histidine 12 that picks off a proton. That sets in motion these electrons being pushed all the way down to our substrate picking up a proton from our other protonated residue. Now, whenever all of that happens, what's going to end up happening is we're going to be breaking a bond within our substrate. And basically, we're going to release half of our substrate after a water molecule rolls in and displaces it. That water molecule that displaces it, it was right here. Okay, could everyone give me a 1 if you are ready for me to kind of not necessarily close the books on this, but transition to now talking about this cell right here. Give me a 1 if you're ready for that, a 2 if you're not, and you have a question about the first cell. Okay, so we've used our base to pick up a proton and effectively lead to breaking a covalent bond within our substrate. Then water rolls in. And if you think back to Gen Chem 2, is water an acid or is it a base? Well, water is kind of, I love using this word, duplicitous. Water can act as an acid under the right circumstances. Water can act as a base, also under the right circumstances. In this case, what we're going to see is basically that. Water is rolling in, it is going to displace a portion of our substrate, the portion that's released. Now, water right here is going to do a pair of things. I've circled my water, and now I'm going to circle the hydrogen, or one of our hydrogens. This histidine right here, the one at the bottom that was previously an acid, it lost its proton. So now it's acting as a base. So it's picking up a proton from water, meaning water is acting as an acid because it's donating that proton. Now, the remaining electrons on water are going to go to help displace and basically prep the remainder of our substrate so that it can donate a proton, or sorry, take off a proton from histidine number 12. So histidine 12 started out as a base, it picked up a proton, and right here, it's protonated. So it's acting as an acid because water is going to roll in, displace a portion of our substrate. It is then going to pick up this proton and return histidine 12 to its state as a base. So when you look at histidine 12 in this first cell, and then in the second, and then in the third, histidine 12 goes from acting as a base to then being protonated and acting as an acid. And then at the end, when it's all said and done, what is histidine 12 acting as? It's a base again. It is deprotonated. Does everyone follow that? If yes, give me a 3 in the chat. If no, give me a negative in the chat. I'm focusing in specifically on his 12. Okay. So now we're going to shift our view down to histidine 119. Well, it starts out as an acid. And then in the second cell here, it's a base. It has to pick up a proton. And then in the bottom cell, it's again an acid. So it goes through this cycling of acid, base, acid. It has to get back to its original state. So if you think about this mechanism, and let's just, I'm going to switch to a white so I can scribble out my substrate. If we think about our enzyme in the starting cell and then in the final cell, well, we had histidine number 12 acting as a base and histidine 119 acting as an acid. In this bottom cell here, what do we have? We have histidine 119 acting as an acid, histidine 12 acting as a base. So in this bottom cell in the lower right-hand corner, my question for you guys is this enzyme ready for more substrate? Give me a Y or N. Yeah. Our two amino acids, histidine 12, histidine 119, they are ready to do this all over again. Now, with that said, now I'm going to ask that same question that I asked a moment ago. Give me a 1 if you feel pretty good about acid-base catalysis and seeing how different residues change. Give me a 3 if you don't feel so good about it. And I'm going to leave out the 5. Okay. And just so you guys know, I do have a video on my YouTube channel where I go over this mechanism. It's my second highest rated video of all time. I think it has 12,000 videos or views, so I'm pretty impressed with that. I'm basically Mr. Beast. Okay, so that's acid-base catalysis. Now, I've got a poll question that I want to do related to acid-base catalysis. What's going on there? And I want to show that to you guys in just a second. Oh, there we go. Poll question is up, and now it's available. Select the true statement about the RNase A mechanism. Mutating to histidine to alanine would not impact this reaction. At the start, one histidine is an acid and one is a base. C, at the start, both histidines are acids, and D, at the start, both histidines are bases. Now, this first point, I'm just going to go into this. I want you to think about it. I like this sort of question because it makes you consider, well, what do I need? What did I see in this mechanism? I saw protons being exchanged. More than anything else, I saw a proton go from here to there. I saw a lone pair of electrons abstract a proton. Um, Alanine. Alanine has an R group that looks like this, CH3. It's a methyl group. How many lone pairs of electrons do you have on a carbon with three hydrogens and going to a carbon? None. So does RNA nuclease have to be made up of histidine? Ooh, Melanie's reading my mind. Okay, so does the RNase A mechanism, one thing that I just want to clear up just so no one else interprets this incorrectly, um, the entire protein's like, I don't know, I think that it's like 250 amino acids long, and it's got every single amino acid in there. But we focused in on these two histidines, which one is an acid and one is a base. So the spoiler alert for the poll question is B. Um, we're focused in only on the active site residues. So if we were to mutate these histidine residues, hist 12 or hist 119, we would need to mutate them to something that can act as an acid or can act as a base. Does that answer your question? Or does that make sense, Melanie? Okay. So if we were to mutate it to alanine, well, that really wouldn't work because alanine, it's not going to push any electrons anywhere. Histidine mutated to, let's say, D, aspartic acid, or maybe a little bit less of an option, but K for lysine. We at least know these two R groups. I didn't mean to spell dork. Well, anyway, I suppose, yeah, nerd, dork. Anyway, um, an aspartic acid or a lysine, those residues can be protonated, and if something can be protonated, it can be deprotonated. Now, histidine's kind of ideal because histidine has a pKa that's like 6.1. So it's like perfect. It's in that good sweet spot. And we're going to see histidine again. Okay, but that's acid-base catalysis. I'm going to end this poll, and the majority of the class did select the right answer of B. One residue acts as an acid, one residue acts as a base. I do want you to like be thoughtful of, be aware of, be thinking of, well, what is an amino acid capable of, basically. Okay, next up is we are talking about covalent catalysis. Now, in covalent catalysis, a covalent bond is transiently or temporarily formed between the substrate and the enzyme or the coenzyme. Now, I want to zoom ahead back to our RNase A mechanism. And I want to stress that, well, in the RNase A mechanism, yeah, the histidine residues and the substrate are close to one another, but they never make contact. There's no bond between them. A proton is taken from the substrate, but that is not a covalent bond. So this is not covalent catalysis. This is purely acid-base catalysis. Now, if we can go back, though, to covalent catalysis, this reaction usually involves a nucleophile on the enzyme. That's important. The nucleophile is typically on the enzyme, and electrophile is on the substrate. So what that will look like from time to time is something like, where did my mouse go? There it is. Well, it looks like something like this, where we have one amino acid Chymotrypsin is an example of an enzyme. And what this has is a residue that has a lone pair of electrons that can pluck off a hydrogen from an amino acid like serine. Serine has that hydroxyl group. And we did mention that serine could potentially act as an acid or a base. But when you see serine, I want you to think, ooh, that's probably going to act as a strong nucleophile as opposed to an acid or a base. Not to say it's off the table, but I want you to lean into the idea that serine is going to help form a covalent bond. So what we have happen here, and we'll get into a mechanism of this in a little while, is that this serine residue loses its proton and immediately forms a covalent bond between that oxygen and part of the substrate. So here is our substrate down here, and we have this serine come down here, finds that partial positive charge on that carbon. This bond right here, lone pairs of electrons, or that shared pair of electrons swing over here, breaking the bond, and here is the temporarily formed covalent bond between part of our substrate and our enzyme. So this carbon right here is our electrophile, our electron-loving group, and then this oxygen right here is our nucleophile, our nucleus or nuclei loving group. Now, some amino acids that are commonly going to act as nucleophiles, well, ROH. Well, what amino acid has ROH? Serine, threonine, tyrosine. What amino acid has a RSH? A sulfhydryl group, or a thiol? Cysteine. What amino acids have amino groups? Well, that would be something like arginine or lysine. And then last but not least, this amino acid or this group can act as a strong nucleophile, and that would be none other than histidine. I switched kind of midway through, going from 3-letter to 1-letter code, so I'll rewrite all of these as 3-letter codes. ARG, LYS, and then HIS. So amino acids. Oh, what is B on the enzyme? Great question. This B is this is another amino acid somewhere on our enzyme that has a lone pair of electrons, so it can act as a base and pluck that hydrogen off. So this serine right here, I said that it was acting as a nucleophile. It's kind of acting as an acid because it gives up this proton to an adjacent amino acid in its same enzyme. But then it acts as a nucleophile to make this bond between itself and the substrate. Does that help? Okay, great. So biologically important nucleophiles are negatively charged and contain unshared electrons. So our good nucleophile form, yeah, serine, threonine, and tyrosine can all act that way because they all have ROH, but only after they have lost that hydrogen are they really going to act as a strong nucleophile, because that's when they have this high density of electrons and the overall negative charge. Now, next up, poll question. Which amino acid do you think would best serve as a nucleophile? I feel like someone's just throwing alanine as their answer or C as their answer because that's what they're doing. Come on, we can do better than that. I've got glycine, serine, alanine, leucine. Let's talk about these. Glycine, that's usually everyone's favorite amino acid to draw because what does the R group look like? Looks like that. Alanine looks like that. Leucine, we've got the chain, CH2, then CH, and then CH3, CH3. Glycine, alanine, and leucine, I'm sorry for saying this word, but they all kind of look like crappy nucleophiles. They don't look like they can do anything. Serine, by comparison, CH2OH. Ooh. I mean, we were already at 81% of the class saying B, but that pushed it up push this up to 83%. We got, oh, 80. Oh, somebody changed theirs for a second. Okay, so the best nucleophile is going to be serine because this hydroxyl group has lone pairs of electrons. So it can actually, one, it can act as that acid to the adjacent residue simultaneously, then it can pick up or it can act as a strong nucleophile. Okay, so now what we're going to do is we're going to jump ahead. And then electrophiles, where are you going to find your electrophile? Your electrophile is going to be found on your substrate. Now, biologically important electrophiles generally are either positively charged or contain unfilled valence electron shells or contain an electropositive atom. Now, I'm jumping ahead a little bit. I've got a couple of different mechanisms in here, and I am going all the way to this mechanism right here. Okay. Now, I'm going to stop the poll because we're done with that and go back. And chymotrypsin is the enzyme that we're going to be looking at with respect to with respect to covalent catalysis. Now, looking at this right here is always kind of intimidating because it's like, oh, cool. Structures, they're close together. Oh man, oh boy. Okay, I'm gonna take a step back because what I want you to know is that chymotrypsin is an example of a protease. Could you give me a one in the chat if you have ever heard the term protease? And any other number if you've never heard protease. Okay, a protease, for those of you who are unfamiliar, a protease is an enzyme that cuts proteins. Whenever I hear protease, for some reason, I always think of cannibal because it cannibalizes those similar to it. It is an enzyme, or it is a polymer of amino acids that cuts polymers of amino acids. Okay, so proteases, that's what they do. Proteases are enzymes that cut proteins. Now, specifically, they are going to cut peptide bonds. Proteases have peptide bonds, but they're going to cut other peptide bonds. Now, the important thing about a protease, and this specific protease is, or the example that we're going to talk about, is a serine protease. There are three amino acids that make up what is known as a catalytic triad. We've got a histidine, and a aspartate, and a serine in the active site of a serine protease. These three amino acids are going to work together. Now, in the next cell, what we're going to do is we're going to look at the individual amino acids and how they change and over the course of these 7 steps. Could everyone give me an emoji in the chat if, okay, cool, I'll follow you along on that journey, is your response. A number in the chat if you say, I'm here, so I'm going along with it. So there we go. Good emoji selection, everyone. I like it. Okay, thank you guys very much for engaging and indulging the emojis. I've seen 2 jack-o'-lanterns. Good job. Good job. Okay, so first things first, like what we did with the RNase mechanism, we wanted to pick out what are the amino acids that are a part of our enzyme, and then what separates that from our substrate. I am going to take each one of these cells on and I'm going to circle our substrate. And this is particularly challenging because, well, we're cutting a peptide bond. So, oh man, our substrate is made up of the same things that our enzyme is made up of. But the good thing for this is that rather than identifying or specifying the amino acids, there's usually like an R group or something like that. So I'm circling our substrate. This is the protein that we are going to be acting on. Now I'm going to be asking for lots of responses. Give me a 9 if you look at this and you say, okay, cool, I can see my substrate. Okay. So now I'm going to erase all my ink. I'm going to start with the amino acid in the lower left-hand corner of each one of these cells. That is aspartate. Now, it's aspartate 102. I don't need you to worry about the numbers or anything like that. But this aspartate residue, let's go through each one of these and say, okay, that is deprotonated. It's negatively charged. Okay, next cell. So after some action has taken place, deprotonated, negatively charged. Third cell, deprotonated, negatively charged. Fourth cell, deprotonated, negatively charged. Fifth cell, deprotonated, negatively charged. Sixth and seventh. All of these, aspartate is deprotonated and it's negatively charged. Now, did anyone see any sort of hydrogen bond, any sort of like ionic bond between that aspartate and anything else in any of these cells? Give me a Y if you saw a bond between that R group and something else, and give me a Z in the chat if you say, I didn't see anything. There wasn't anything. You're right. Now, if you have ever seen the movie The Big Lebowski, add an L to the chat. Okay. The entire premise of that movie is it has to do with furniture. The movie's about furniture. The movie is about a rug that tied the room together. This aspartate right here is the Lebowski's rug. This aspartate does nothing other than occupy space because that's one of the important elements of an enzyme's active site. It's there so that all the other amino acids are like, okay, well, I can't go over there because that's where aspartate is. Now, with that in mind, if this aspartate were to be mutated to something else, to say like a histidine or a leucine or something like that, or a glycine, those are smaller residues. The space that's occupied is going to be different, in which case the reaction is going to be a little bit different. Now, what I want you to take away from that is that aspartate really is there for facilitation. It's kind of like it's a chaperone at the junior high school dance. It's there so that, there's no fights and nobody throws any Coca-Colas or anything like that. Okay, so how does everyone feel about that aspartate? Give me a 1 if you say, okay, I'm good, and a 5 if you say, I feel like I'm good. I feel like it should be doing something, but it doesn't appear to be doing anything, which is kind of like the 1. Okay, so what I'm going to do is ask you to just kind of ignore the aspartate for the time being and just know, like, okay, I'm going to file it away because it's there. But in terms of pushing electrons, aspartate is not doing anything. Now we're going to switch to our serine. Serine. What do we know about serine? Well, it can act as a nucleophile. It can act as a good nucleophile. In order to act as a good nucleophile, it has to lose a proton. It has to have that lone pair of electrons or those lone pairs of electrons in order to make a bond. Serine over here, okay? It's got the hydroxyl group. Next cell, boom, that hydrogen's gone. Not only that, but that serine has a bond going up to our substrate. Third cell, same sort of thing. And oh man, our bond with our substrate gets pretty complicated. Fourth, same. And oh, our substrate's changed. Fifth, sixth. Oh, it seems to me like that serine looks like it's pretty important for establishing a covalent bond between our enzyme and our substrate. So serine, what has to happen for it is it gets deprotonated. And when it gets deprotonated, it's going to be able to make that covalent bond with our substrate. At the end, down here, it's going to return to its original state, where it does not have that covalent bond to our substrate. Give me a 1 in the chat if you say, okay, cool, I can follow what serine does. It loses a proton, makes a covalent bond, stays, loses the covalent bond, and now is protonated again. Okay. So what I'm wanting us to do is walk away from this with an idea of like, okay, well, all of these things work in concert with one another. I know what each one of them does, and I know how they work as a like interconnected system. The last amino acid that we're going to talk about in this mechanism is histidine 57. This histidine right here, is it protonated or deprotonated? D for deprotonated, P for protonated. Perfect, it's deprotonated. So if a histidine residue is deprotonated, what is it capable of doing? Picking up a proton. Do we know of an amino acid that we've talked about so far that would love to give up a proton? Yeah, serine. They're right next to each other. And that right next to each other is not simply for descriptive purposes. If you were to shrink yourself down to the atomic level and get into the active site of this enzyme, you would see that serine is right here, that histidine is right here. They are next to each other. They are neighbors. So that this proton or that this histidine can abstract this proton very easily. That deprotonated histidine picks up that hydrogen, making our protonated histidine. In our next cell, oh man, histidine goes from being protonated to deprotonated. What? Okay. Well, this is a place where we have to look up to our substrate and say, oh, you know what? Our substrate down here or our substrate there, it's going to use a pair or a lone pair of electrons because it's still in close enough proximity to pick up this hydrogen. And whenever it does that, whenever it picks up that proton, our histidine goes from being deprotonated to protonated to deprotonated and stays there for a while. But that proton, so histidine acts as a base, then it acts as an acid, and that enables our our substrate to basically break off. Then, with the help of H2O, which can act as an acid or it can act as a base, we are able to donate our proton from water onto histidine. Our protonated histidine is now an acid. It can donate that proton back to serine, which enables our shared pair of electrons in that covalent bond from serine to return from whence it came. Now, if you're thinking about this and you're saying, I felt really good about aspartate, I felt really good about serine, and then histidine just kind of came in and was like, I'm doing this, now I'm doing this, now I'm doing this, and then here, oh, the reaction's done. What I want you guys to know is I want you to be able to answer the question of which amino acid forms the covalent bond with our substrate. Then I want you to be able to answer the question, if I were to show you, if I were to grab one of these cells, if I were to select this right here and ask you the question, well, what is histidine ready to do? Or what is histidine capable of in this cell? I'd want you to be able to answer that question. I'm not going to look for a long dramatic response and explanation that, well, histidine, it sets out on a journey to find itself. And whenever it does, the first strife that it encounters, it becomes an acid. Then it turns to the west and becomes a base. I'm not looking for that. Beautiful story, very poetic, very valuable. But what we are most interested in at this point in time is, well, serine, what is it doing? It's establishing the covalent bond. How does that happen? Well, with the help of histidine. Then how do we get back to our original serine? Well, again, with the help of histidine. Basically, between cells 3 to 6, histidine goes through lots of changes, which are a result of lots of other parts of this mechanism changing. But what I'm most interested in you knowing about is if you could quickly summarize each residue. Aspartate, it's important for space. Histidine, it's important for acting as an acid and a base. Serine, it's important for acting as a nucleophile. How does everyone feel about those three descriptions? Give me a one if you say, okay, I got you there. And then a three if you say, that's, I, I'm still struggling with it. Sure. So I'll write it down. ASP, I'm going to say spatial awareness. Histidine, acid, base. And then serine, nucleophile. I'm going to say nucleophile plus um, covalent bond with substrate. That's a pretty succinct explanation of all of them. Now, my follow-up to that is with those three descriptions, can you see that in these different cells? Give in this whole cell. Give me a one if you say, yeah, I can see all those, and then a nine if you say, I see one of them, but not all of them. Okay. Okay. So this was our second type of catalysis. This is covalent catalysis. And if you're thinking, but histidine, that's an acid-base catalysis. You're right. Histidine does act as an acid base, but you can kind of think about this as, well, as soon as we had that covalent bond formed, that usurped or that became kind of a higher priority for explaining this. When we had a covalent bond established, now all of a sudden we we're only focused on, yeah, this is covalent mechanism. Okay, so the last type of catalysis that we're going to talk about, I gotta come, there's some other stuff. The last type of catalysis that we're going to talk about is metal ion catalysis. Nearly one-third of all known enzymes require the presence of metals for catalytic activity, which if you think about this, are we talking about a neutral metal or are we talking about a metal ion? We're talking about a metal ion. We're talking about something that can, adopt a couple of different oxidation states, something that can pull electrons in one direction, maybe release electrons in another direction. For this, we have metalloenzymes. Metalloenzymes are enzymes that tightly bind metal cofactors like iron 2, iron 3, copper 2, zinc 2, manganese, cobalt 2. There are also metal activated enzymes. And this has to do with basically how tightly does that enzyme bind. If an enzyme loosely binds to a metal, then that would be classified as a sorry, if an enzyme loosely binds a metal, that is a metal activated enzyme. If it tightly binds it, it is a metalloenzyme. These two terms are not interchangeable, though you maybe have heard them or maybe somebody has used them interchangeably. Now, a cofactor is generally considered a metal ion. A coenzyme is generally an organic molecule like NAD. Could anyone remember from our discussion of enzymes, do you remember the number or the EC number that corresponded to NAD plus or enzymes that use NAD plus? So remember we had our oxidoreductases. Oxidoreductases were one, and we had that glyceraldehyde 3-phosphate dehydrogenase, or GAPDH, is an enzyme that uses that. And so NAD plus loves to grab onto electrons and hold on to them. NAD plus is a coenzyme. Now, when we talk about metal ions, we use these two different terms, apo and halo. An apoenzyme is an inactive form of an enzyme in the absence of its cofactor. A haloenzyme is the active form of the enzyme and has that cofactor or that metal ion present. Okay, now for metal ion catalysis, sorry, I was collecting my thoughts. I know that it might have looked like I was about to start crying. I wasn't, and I'm not. Um, but it's okay if people cry. Carbonic anhydrase catalyzes a pretty simple reaction. CO2, H2O, makes HCO3, and H plus. We talked about this enzyme. We talked about it in the context of cellular respiration. Every time that your body breaks down sugars, one of the products is going to be CO2. CO2 is a waste product. You have to get rid of it. Well, how does your body do that? Well, your body does it a couple of different ways. One way is that it uses hemoglobin and basically just anchors a CO2 molecule on top of it. Another way is with this enzyme carbonic anhydrase. It takes CO2, reacts it with water, and makes this HCO3 minus ion. That ion can go into solution more easily than, say, CO2. CO2 is pretty insoluble in water, which is why, or within an aqueous solution, which is why you have carbonated beverages, that there's bubbles of CO2 that emerge. Okay, that's what this reaction is. Now, when you look at an enzyme catalyzed reaction, I mentioned that it's important to be able to pick out the substrates and pick out the enzyme. In this model, we've highlighted 3 histidine residues. Those 3 histidines are our active site histidines. Now, what they will do is they are positioned in a way to basically just hold on to a zinc ion. So histidine, those are active site residues. Now, looking back at our metal ion catalysis definitions, metalloenzyme or a metal activated enzyme, carbonic anhydrase is an example of a metalloenzyme. So what that means is that these histidine residues, these 3 histidines are positioned and they hold that zinc 2 plus in place. And it's almost like they're holding on to it, waiting for something that's kind of negatively charged to come around. Now, these histidine can be protonated, it can be deprotonated. These residues, chances are, those are going to be deprotonated. They're going to have some lone pairs of electrons that attract that zinc 2 plus. That zinc 2 plus itself is going to be held in position to attract something with lone pairs of electrons. Lo and behold, H2O rolls up. H2O has lone pairs of electrons. It's going to grab onto that and hold on to it. And whenever it does, could anyone add to the chat, especially if you haven't looked at this slide before, or if you have, it doesn't really matter. Could anyone add to the chat, what's the pKa of water typically, or roughly? Yeah, about 14. Now, what this is going to do is this water-zinc interaction right here, it's going to cause the pKa of water to go from 14 down to 7. So what that means is that this water molecule, let's say that you had multiple copies of this whole thing right here. Let's say you had 10 copies of it at a pH of 7. Well, 5 of them are going to be deprotonated and 5 of them are going to be protonated. So 5 will look like this and 5 will lose one of those protons because the pKa has dropped. Now, the key there, your important takeaway is that the pKa dropping, pKa going down, causes this proton to nope out of there. So what we're left behind with is this zinc 2 plus ion with a hydroxide ion, that OH minus. And that is when our CO2 rolls up and says, hey guys, what's going on? And just as the CO2's ready to have fun, party with his pals, that's when some of these lone pairs of electrons that make up this negatively charged hydroxide swing over to the carbon and say, get out of here. And then this shared pair of electrons swing down to that oxygen, which temporarily stabilizes it, but at the end of the day produces a molecule of none other than HCO3 minus. That HCO3 minus is then more soluble, and so it is displaced by H2O, which, heck, is there enough water in the environment? Yeah, there's plenty of H2O. That HCO3 is displaced, we get another water molecule, and then this process starts all over again. Now, let me ask you this. I mentioned that these histidines more than likely were deprotonated. Did I ever go back to talk about those histidines again? Y or N? No. Did the histidines protonation state ever change? Do you think in this mechanism? No. As long as those histidines hold on that zinc ion, well, they're good. They're fine. If the environmental pH changed radically, would they change? Yeah. Would this enzyme lose some activity? Yeah. But that's not what we're focused on because how are these electrons being pushed around? Well, they're not being pushed around with the assistance of any sort of amino acid intervention. These electrons are being pushed around solely because of this zinc ion that's held in place. If these histidines lost that zinc, would this reaction still take place? No. The amino acids are kind of like the conduit that hold on to the zinc that does the reaction. And I apologize if I didn't use the word conduit right. At any rate. Okay. Does that make sense? Could everyone or give me a five in the chat if okay, I follow that, or a T in the chat if you want me to go over this again. Okay. Okay, I've got another. Oh, there's my mouse. Okay, there we go. All right. Now I'm going to launch a poll question. We're going to go over it kind of together. If histidine is at the center of carbonic anhydrase mechanism, what property of histidine makes it particularly valuable for this reaction? Its pK of around 6, making it capable of being protonated or deprotonated at a neutral pH, having a ring structure, having a nitrogen in its ring, or D, none of these. Histidine is not important for this reaction. Well, I'm going to go ahead and say D is not the answer. A is kind of interesting. It's kind of tempting, but this is really just comparing protonated versus deprotonated. And we didn't see the protonation state of histidine change. Histidine having a ring, maybe, but that seems a little bit abstract. So let's compare B and C. Histidine having a nitrogen in its ring. Let's think about nitrogen. Nitrogen with 1, 2, 3 bonds. What else is it going to have? Lone pair of electrons. Ooh, it's kind of interesting. So if you were to compare C and B, you could have a ring with oxygens in it, a ring with just carbons. The ring structure itself is not, I like this word, intrinsically important or interesting. So now we're kind of back to A and C, and answer option A mentions the protonation state. Yeah, that's valuable to know that it's deprotonated, but the fact that it oscillates or changes between being protonated or deprotonated isn't really impactful for this reaction. So really the best answer for this question would be C, having a nitrogen within its ring. Now, does it have to be a ring structure? No. But having a nitrogen that is deprotonated with those lone pair of electrons to attract a zinc ion is what makes this compelling and interesting. Okay. Now, the next mechanism that I'm going to go over, actually, I'm going to skip over this lysozyme mechanism. Well, I'll touch on it very briefly. Lysozyme is an enzyme that breaks cell walls, hydrolyzes the peptidoglycan layer of bacterial cell walls. It breaks these glycosidic linkages, beta 1,4 glycosidic linkages. Those are the bonds between sugars. So it's a beta linkage between this sugar and this sugar, N-acetyl-muramate and N-acetylglucosamine. Okay, cool. Now, what I really like this picture right here because I want to draw your attention to a couple of amino acids, aspartate 52 and glutamic acid 53. Those amino acids are pretty central to the catalysis of this enzyme. Now, a fair question then is, well, why are we looking at aspartate 101, tryptophan, alanine, asparagine? Wait a minute, alanine, what the heck? You said alanine doesn't do anything. I stand by that. Alanine doesn't do anything, really, in terms of catalysis. But let's take a break for a second. Is this the alanine R group? Um, is that the R group for alanine? Give me a Y or N in the chat. No. Could anyone add to the chat what that is? And I'm going to circle some other examples of the same thing, like glutamine 57, glutamic acid 35, phenylalanine 34. What part of a protein or of an enzyme has this right here? That's a carbonyl oxygen. What I'm getting at is this is the backbone. Could you give me a... Oh, perfect. Thank you, Melanie. Absolutely. The backbone. Yeah, the backbone's important for secondary structure stability, but the backbone is also involved in not necessarily catalysis by breaking and making bonds, but all of these other residues. I'm going to scribble out the aspartate 52 and glutamate 35. All of these other residues, they're very much like the aspartate that we looked at in the serine protease mechanism, the chymotrypsin mechanism. What they do is they help position the substrate in a proper fashion so we can break the bonds in it. So that's why you'll see a mechanism, you'll see a bunch of residues that you first are like, they don't have anything to do with this. They might not. But do we only care about active site residues? No. We care about other parts of the protein because other parts of the protein contribute to stability of the protein. They contribute to positioning of the substrate properly. Now, looking at the specific reaction, the two active site residues in the lysozyme mechanism are glutamate 35 and aspartate 52. Now, I like to show you this because my first thought whenever I see this is, well, glutamate 35 and aspartate 52, if I see those two residues, oh, acid-base catalysis, done. That's all there is to it. Well, if we look at this glutamic acid 35, sure enough, our substrate, which is in the middle here, our substrate is using this glycosidic linkage to target that hydrogen. So could you tell me what glutamate 35 is doing? Is it acting as an acid or a base? Just from this first cell? Well, it's acting as an acid. It's donating that proton. It is protonated, so it can donate that proton. Okay, so then a fair question is, acts as acid. Aspartate 52, you look down at that and say, oh, well, it's deprotonated. It's got to be acting as a base. Well, let's take a look. Okay, aspartate 52, this is our first shot of it. Then our next shot, we've got, it's negatively charged, and it's swinging up to, wait a minute. That carbon, wait, a bond between carbon and oxygen? That sounds to me like that would be a covalent bond. So we have this mechanism where we've got general acid catalysis because of our glutamate 35. Then we've got covalent catalysis because of our aspartate 52. We have that aspartate 52 that holds on to our substrate because half of our substrate's been broken off. The important bond that we wanted to break has been broken. Then what we're going to do is bring water in so that we can disrupt this. Now, and then at the end of the day, that water is going to act as an acid because glutamate 35 is acting as a base. It's going to pick up that proton. We've got our protonated glutamate 35. What I want you to take away from all of this is that just because you look at something and say, oh, there's an aspartate, it's got to be acting as an acid. Or, oh, there's a glutamate, it's got to be acting as a base or something like that. Look at the steps a little bit carefully and say, okay, there's no proton being exchanged there. That's not acid-base catalysis. Could you give me a one in the chat if you say, okay, cool, I follow that. All right. Now, I'm going to go into a little bit more about proteases. Proteases, as I said, they are proteins that cut proteins. They're enzymes that cut proteins. They cut peptide bonds. You need at least a dipeptide to do it. Serine proteases, what do they have? They have a serine in the active site. We saw the serine in our chymotrypsin example where it formed the covalent bond. Chymotrypsin and subtilisin are two examples of serine proteases. Thrombin is a clotting enzyme or is an enzyme involved in clotting. Oh, great question, Melanie. I'm sorry, I blew straight past that. This is not as simple or as trivial as acid-base catalysis. Because of the presence of this covalent catalysis, we would refer to this mechanism as a covalent catalysis mechanism. It's a great question. Okay. Or you'd say it's some mixture of various types of mechanisms. Now, for you guys, what I would do is show you a step like this. I'd capture that whole thing and say, what is, again, what is aspartate, or sorry, what is glutamate 35 doing? It's acting as an acid. Okay. What about aspartate 52? Well, it's acting as a nucleophile to establish a covalent bond. Those are the sorts of questions that I would ask. Okay, so chymotrypsin, subtilisin, and thrombin are all examples of proteases. Trypsin, chymotrypsin, elastase are digestive enzymes. Subtilisin is a bacterial protease. Plasmin breaks down the fibrin polymers of blood clots. If anyone's planning on going to medical school, if you've ever heard of, or if you know of like the most immediate and urgent response for someone having a stroke, does anyone know what that is? I'll give you a hint. It's three little letters. Well, not little letters, three letters. It is TPA. It's tissue plasmin or plasminogen activator. Now, plasmin is an example of a protease, and it breaks down polymers of clots. TPA is also known as the clot. Every time I say that, I say clot bluster. Clot buster. If someone has a blood clot in their brain or something like that, and they're administered TPA within, I think, a 3-hour window of whenever the clotting began, they will have, or their symptoms of their stroke are pretty minor because that clot gets busted up. Tissue plasminogen activator, or TPA, cleaves the proenzyme plasminogen, yielding plasmin. What on earth? Proenzyme plasminogen? Yes. Okay. A zymogen. A zymogen is a molecule or a protein that floats around. It doesn't do anything. It's inactive. This is a molecule or a protein that is activated by proteolytic cleavage. So, do you want an enzyme just floating around willy-nilly cutting up every protein? No, you don't. You want a protease to be active when it needs to be active. And so this enzyme right here, chymotrypsinogen, inactive. But another protease known as trypsin will come along and cut two places. And whenever it does that, what it makes is something known as pi-chymotrypsin. Trypsin will cut in one location. That pi-chymotrypsin cut by trypsin will then cut itself to yield alpha-chymotrypsin, the fully active form of it. The way that I always kind of think about this is a protease is something that floats around and is ready when it's called upon. And when it's called upon, it will be activated by something else. So a zymogen is an inactive enzyme. But then whenever it's cleaved, it will be activated, and then it can go on and cut up any and every peptide bond that it encounters. So yeah, there's that. Now, whenever I say they can cut up anything that they want, that's a little bit too simplistic. The last thing that I'm saying today is on this slide, and that is that zymogens are inactive, but activated by other proteases. So with that this is one of the things that's kind of weird. A protease is inactive until another protease wakes it up and activates it. So a zymogen is an inactive protease. Okay, that's where I'm going to stop. We got our poll questions. If anyone has any questions, you are more than welcome to hang around. There's some stuff on this slide deck that we're going to revisit later on. Other than that, I hope you guys have a great weekend. And I will see you on Tuesday. And your exam grades, I am working on them and I hope to have them done by the end of the day. I apologize for the delay. I will get them out to you and release the questions. Did you do a lecture on the day one slides? From week 8? Yes, I did that last Thursday and I did it on Tuesday.